the publication of Biological Science, Exploring the Science of Life. So I'm John Crowe, Editor-in-Chief for Natural and Social Sciences in the Higher Ed Department at Oxford University Press. I'm delighted to be joined today by two of the authors on our Biological Science project, Dr. Dawn Hawkins from Anglia Ruskin University and Dr. Jenny Koenig from the University of Nottingham. So the focus of today's webinar is on the centrality of quantitative skills to the teaching and understanding of biological science, the challenges often faced in teaching quantitative skills to biological science students, and approaches that can be taken to mitigate for those challenges. And I can't think of two better people to speak to all of this and to have contributed to our project than Dawn and Jenny. So I've known Dawn for almost my entire publishing career. In fact, she was one of the first authors I ever commissioned to write for me at OUP. And in the intervening 20-odd years, I've been in awe of her unwavering commitment to teaching quantitative skills and how important those skills are to success as a biological science student. And Dawn and Jenny's collaboration has been equally long-standing, so it felt only natural for Jenny to work alongside Dawn on our project, and their expertise complements each other perfectly. And in particular, Jenny has extensive expertise of teaching maths pre-university, and that experience really has been invaluable when it comes to our project being realistic about what students know and understand when they start their undergrad studies, and also where extra support is likely to make all the difference. So thank you to Dawn and to Jenny for being here today. That's enough from me for now, but before I hand over to Dawn, just a couple of things. Firstly, do please use the chat function to contribute to the discussion throughout this session. And we'll also have time for a Q&A session a bit later too, so please use the Q&A panel next to the chat to submit any questions you'd like Dawn and Jenny to respond to. And I'll be joined by my colleague Stephanie later in the session, who will facilitate that Q&A. But for now, over to Dawn.
Hi, great. So that was a very nice introduction. Thank you, John. I didn't really, though, know we'd been quite that long-standing, but yeah, it goes back a long way. I haven't known Jenny for quite as long, but I thought I would start with, it's been quite a while, and I thought that's actually quite an insight, give you some insight into where we're coming from. I just sort of describe how we met and the sort of experiences we've had. So the first time Jenny and I met was in Reading in 2010. It was at a workshop being held by the BBSRC and the ATA on challenges for math challenges for biologists. And although we both worked and lived in Cambridge, this was the first time we'd actually met. And it was the start of where we realised that though we came from quite different perspectives, we had very similar experience of the problems and ideas about solutions. So Jenny was teaching maths in a more biochemical setting at a Russell Group university, and I was teaching stats to whole organism biologists at a post-92 university. But despite that quite different backgrounds and approaches, we actually had very easy to see that our commonality of our experiences of the challenges that we faced teaching biological maths or stats to, in my case stats and in Jenny's case maths, to biology students. And actually at this conference it was evident that, you know, we weren't the only ones, it was a widespread problem. And indeed, leaping on from there, it was widespread amongst different institutions and indeed internationally, particularly Jenny has found as in reports she's done, that these issues are actually not confined to the UK. She's looked at in Australia and in the US, and similar challenges have been found. And so why that is and how to solve that was something that we talked about quite a lot. And we got invited to various institutions to contribute to discussions on policy and curriculum development. We've talked at the Nuffield Foundation, the Royal Society of Biology, the British Academy, the Royal Society of Biology, the Royal Statistical Society, the Wellcome Foundation. Those are the ones off the top of my head. They were, because we've been successful in our own experiences of teaching this, and it's been picked up in various ways, then we were being asked to, you know, contribute to this the problem being more generally recognised, and we were being asked to contribute to that. So I think things have improved in some areas, but we still face quite a few challenges. And I think what we've done in the toolkit is brought our experience of those challenges and how we've solved them and how we found ways to solve them, and how being part of this network of people and institutions and hearing other people's solutions, those solutions we've applied to the toolkit. So we've also, Jenny has experience of teaching in schools and was actually sat on the Department of Environment Maths expert panel for core maths. So we've actually had reach out from the maths community to biology. We've been points of contact because mathematicians are wanting to know, how we can make that connection and how they can best prepare students for their more applied areas. And also, I feel that in the stats area, there's been an element of me being able to say, look, statisticians, actually, I can remember saying this at the Royal Statistical Society, you can use biological scenarios to make your teaching of statistics more interesting. So I don't think it's been a one-way thing. I certainly, because everyone, lots of people are interested in wildlife and those what they've had for films, and if you can set them in that context, you can perhaps grab people's attention. So it's been both ways as well. We've talked to people on A-level exam boards and been invited to various universities. Dundee and Hertfordshire come to my mind, the ones I've talked at, and various conferences. And this is again because people had problems and wanted to reach out and understand, wanted to connect with people that had solved this or partially solved it. So we've also been invited to talk at things like the Biochemistry Society. Jenny, I mentioned already, had produced a couple of reports on, which were quite early on, actually, in 2011 and 2013 with the HEA, again, highlighting this problem and going forward. So it's not a new problem. It's not a problem to a particular institution or a particular subset of biology or type of, it's ongoing. And so there have been some changes. There have been some changes in the math curriculum, and we've been to support that. But we feel that there are still challenges there. And actually, I can see that the poll is open, and if you, I can already see that on the poll it says what challenges, if any, have you experienced teaching biological students quantitative skills. So this is a poll. I think you can all see a poll with that question on. I can see that we haven't had anyone pick the none yet, so it's still clearly somewhat an issue. And we might pick on some of that later. Finally, what it's been able to me to do is to write textbooks, of course, and John has described that he was the person who commissioned one of the first books he commissioned, and it was because he came to our institution and saw that we, not just me, actually a team of us, had actually solved, or at least addressed in a large part the problems we were having with teaching stats to our students. And that's when my book called Biomeasurement got commissioned, and that's what led to me contributing now to the quantitative cause. And it was great that Jenny would come on and service the math side of it. Anyway, I think, I was going to say, the key approaches, what we've actually done is actually, I think, putting things in a biological context is really important. That's what we've done. So putting things in a biological context, and for me, talking about the data and understanding the data for the stat side, it's more about understanding the data than the nitty-gritty of the techniques and the underlying process. I think that's what we've headed for in the quantitative core kit. But I'm going to hand over to Jenny, who's going to talk more extensively about the challenges in relation and solutions in relation to math. Over to you, Jenny.
Okay, thanks, Dawn. So I guess, yeah, from what Dawn was saying, starting with that biological context is important. But I think the other thing it's been really fascinating actually working with Dawn is that we've been working across the whole of biology, and that's proven to be actually quite challenging because I'm used to the biomedical end of things and the very molecular end of things, and quite a lot of the time the maths is the same. The maths that underpins is just the same, but there may be different terminology used. and that's that can be difficult when you're used to working in a particular area and used to using certain symbols, and then you're having to teach across the whole of the biology. So that idea of not only the symbols but also the language. So there's a few things that we wanted to pick up on today. It sort of distills down, I guess, into three areas. One is language and how we use language in maths teaching and maths learning, actually. The other one is visualization. So this is something I've discovered that is a particular thing of mine, that I visualize things very strongly. So I'm going to show you some of these ideas for helping students to visualize what's going on with the maths. And then the other thing is pitching it appropriately. So we made the conscious decision early on that we were going to have in mind the student with no A-level maths, no calculus, no logarithms, and have that student who's got a GCSE around about a grade 4 or 5, which would have been B or C in the old system. And we chose that basically from a number of the surveys and reports that we'd done previously. It seems to catch the vast majority of biological sciences students. So I'm just going to talk through a few of the ideas and give you a few examples from a few screen grabs from the book. But the first thing really is this idea of catching these gaps. What is the assumed knowledge that we assume they know, and what is the actual knowledge that they actually have? So what are the common issues? And there are quite a few, and we try to make a point of pointing these out really clearly in the text. So, for example, using litres rather than decimeter cubed, and milliliters instead of centimeters cubed. For some students, that's obvious, but actually for a lot of them, they can be a little bit confused. Capital M for molar concentration, letting them know that that stands for moles per litre. That was a bit of a eureka moment for me at one point, because they will have seen that as capital M for molar mass or relative molar mass, rather than as moles per litre. In biology, we use some really wacky units for concentration, so percent weight per volume, parts per million. They may have seen those things before, but not really know what they are. And the other thing is negative powers, numbers with negative powers. So a lot of students struggle, certainly when I was teaching at secondary, students struggle when you get to squares and cubes, and then having positive powers, they sort of can get there. But when they're presented with negative powers, I think they struggle to have a sense of number around negative powers. There's a few symbols, things like the sigma symbol for sum and the delta for change in, and just sort of being really clear about what those things mean. So what stemmed really from the idea that we were starting from the point of a student with a GCSE around about a 4 or 5 grade, means that we've not included any calculus. Having said that, there is a toolkit on rates of change. And anyone who's done calculus, if you look at that toolkit, whilst there is no calculus in there at all the phrasing, all the language, all the words, all the way of it, you could almost have calculus come next, if you like, but not in this text. So I'm just going to share my screen. Please do ask if there's, or put some questions in the, I can't talk and share my screen at the same time. Okay. So hopefully you're seeing one example. So one of the things we did decide we needed to do was cover what logarithms are. So logs are not in GCSE maths. They are in A-level maths, but only a minority of biological sciences students will have done A-level maths. So what we're doing here is sort of recognizing that the notation for logarithms is a bit cumbersome, and trying to explain that the log of a number, so the log of 100, you can work that out by expressing 100 as 10 to a power, so 10 to the 2. So the log is the power there in red. The log is the power to which the base is raised. So the log is 2 and the base is 10. Another thing that we're trying to do is use this idea of visualizing concepts. So linking macroscopic, microscopic, graphical and symbolic representations, and then pulling that together with a prose description of the biology and how this all fits together. So this is sort of an extension of Johnson's triangle, if you're familiar with Johnson's triangle from chemistry teaching. This extends it through into the mathematical symbols and the graphs. So looking at the colored solution and at the macroscopic level, and then prompting them to think about what does that mean for the number of molecules in that solution, colored molecules, and then linking that to what you measure, the absorbance, and then linking that to the symbols in the equation, and then pulling all of that together with some words that they can read. Developing number sense is also really important, and getting the hang of this index notation, which for a lot of students is not always comfortable, and linking together what a negative power actually means with a sort of visual representation like this. So this is in the book, this is in the context of bacterial growth or cell growth, which is why we've got things to the base 2. But there's an equivalent diagram with numbers to base 10 as well. So showing that something that has a negative power is representing a fraction. So 2 to the minus 2 is 1 over 2 to the 2. So as you get the power to be a bigger number and more negative, then the overall number is getting smaller and smaller and smaller and smaller. So developing that number sense through sort of visual diagrams, but also through questions. So here's an example. We've got solutions, filter to remove bacteria. There's the pore size of the filter. And then we've got a bacterium, and then we've got a virus. And getting them to apply that idea and think about which of those numbers is larger and which is smaller. Sometimes there's a misconception or a misunderstanding that if the number is big, if the power isn't a bigger number, that means it's a bigger number altogether. So they sometimes think 10 to the minus 7 is bigger than 10 to the minus 6. So you really got to point out that it's not, and try and pull it all together and explain why. Another one, trying to get more fluency with handling these numbers with negative powers. So putting in prefixes, so like nano in this case. So taking 3 by 10 to the minus 8 meters, changing that to 30 by 10 to the minus 9, and then putting in the nano for the 10 to the minus 9. So sort of scaffolding that calculation, providing a way to think about that. It's really common that they'll go in the wrong direction. So instead of 10 to the minus 8 to 10 to the minus 9, they'll go to 10 to minus 7, for example, and then get confused. So scaffolding the calculation in several steps can also help to just avoid those mistakes, I guess. What else? Oh, yeah, language. So, yeah, how to read and say the maths. So we know from the maths education literature that people don't read equations. Their eye skips past. They read the words, they skip past the equation, and then they read the words again. So you've got to say what the equation is saying in words as well. But the same applies to fractions and things. So if we talk about 1 in 6 or 1 out of 6, those words typically mean 1/6 as a fraction, and pointing out that it's 1 divided by 6, 1 out of 6. And the same is, when you've got grams per litre, if you've got 9 grams per litre, making that link. And I guess, I suppose the thing I find difficult is when you're used to this and you're familiar with it, it sort of seems obvious. But when it's new, it's not obvious at all. So it's sort of taking yourself back into that previous life when you were that student trying to get your head around all of this. Another way of visualizing concepts and scaffolding is using this bar model. And this was really transformative to me when I started teaching in secondary, and I just wanted to share this example. So when we look here, we've got on the left, a part-to-part ratio. That's what it's called. So, for example, fertilizer mixture N to P to K of 5 to 7 to 4. So notice I'm saying 5 to 7 to 4, and pointing out that that's how it's said if you were going to read it and say it out loud. And this question actually came from my daughter, who's studying environmental science. So she said, "Mum, I don't know how to do this." And she was given, total weight is 500 grams. How much phosphorus will it contain? And all I did was draw 5 boxes to represent the 5 parts, and then 7 boxes to represent the 7 parts, and then 4 boxes to represent the 4 parts. And I said, well, altogether, that's 500 grams, isn't it? And she said straight away, oh, yeah, I know what to do now. And I didn't even have to say, I was like, oh, okay, well, good. So visualizing the calculation in that way helps you to sort of think through what it is you need to do then it becomes much easier. I think one of the things that is difficult about dilutions is that a 1 in 5 dilution, we say 1 in 5, that's actually a part to whole ratio. We're talking about a 1 in 5 rather than a 1 to 5. So being really clear about that language. So a 1 in 5 dilution is actually a 1 to 4 dilution. And then if you set out your calculations like I've done there, lines one above the other, that just helps to, it helps to visualize what's going on, and it helps to sort of set it out in steps. And then it can help the thinking through. It can support the mathematical reasoning, I guess. That's what I'm saying. So there are some things that don't translate well in text, and for that we've used video. So for example, rearranging equations. So I've chosen one of the more complex ones here, which is rearranging the Michaelis-Menten equation. So there is a simpler, earlier one, which rearranging concentration as amount over volume. But in this case, showing that on a page, it's just a whole jumble of symbols. So the video is really there to take you through it step by step, and just sort of reason through and explain. So it's talking as I'm writing and explaining what I'm doing and working it through. So sort of modeling that behavior. The other thing we've done is using spreadsheets as a tool for 2 things, really. In this case, we're looking at doing calculations. So how to use a spreadsheet and typing in formulas, and getting it to do, in this case, the Shannon-Vrena diversity index. But sort of taking people through how to set it up so that no matter what calculation they're doing, they should be able to apply the same principles. And then the other one is for visualizing equations. So here we've got a spreadsheet that's been set up so that it displays the graphs, and they just change. So you can change the values in those yellow cells, and then you can see the shape of the graph change. So then you can get a better feel for, well, what happens if I change A, and what happens if I change B, and what happens if you put it on a log scale instead of a linear scale for the x-axis, and all of those. You can sort of play with it and get before I was talking about number sense, now I'm sort of talking about equation sense. Looking at the symbols in that equation and getting a sense of what it's telling you. It's a description that you should develop an ability to picture and visualize what it would look like if it was graphed. The other thing is effective use of units. So this can be really helpful for students to make sure they have the units in the calculation with the numbers, and ensure that the units cancel out. This is probably the most complex example that we go into, Fick's law, where you're given a whole bunch of different parameters and you have to put them into the equation, but you've got to do lots of playing about with the units first to get them in the right units, so that then when they go into the equation, they all cancel out nicely. And if you notice top left there, there's a diagram that's sort of trying to get across what's going on here. So it's not just a page with maths all over it. When they first see this, it's just a page with biology there and a few numbers in the bottom left. And then gradually as I talk, I'm writing more and more onto the screen and gradually building it up. So it's not too scary to start off with. And okay, so that's just a few things that are about the math. So if I hand over back to Dawn.
Leave it up. I think it's easier if you just leave it up or save some time.
Yeah. Okay.
So, yeah, so things come out. So things we've learned from other people, things we've learned from teaching ourselves. Language is really important, as Jenny has shown with I think that ratio example is really, that when Jenny wrote that part of the textbook, that quantitative skills, I read that, wow, yeah. So language is really important. Visualization is really important. Getting a summary. This is so Jenny has shown you with the math side. So this is an example of visualization with the stats side, visualizing the different levels of measurement, different types of measurement. Students come with this idea that data can only be sort of, basically they're visualizing measures, scales, weights of babies, heights of trees, but data can be of different sorts, as we all know. So, and that actually is quite, it's very simple, really, but it's actually really important because ultimately when you go on to decide between a Q-test and a Man-Whitney U-test, whether the data is scale, level, or ordinal is what makes the difference. The structure of the data, things like what sort of design. If you've got a repeated measures design, then you're talking the difference between, if you're comparing two samples, a Q-test and a paired Q-test. So that very simple, you can start to see it, just understanding about the data determines what kind of analysis you use, even down to obviously, which type of central tendency you use. So that foundation and that's part of the stat side, and this is one of the visualizations of that. Next slide, please, Jenny. And sort of picking up on things like Jenny's, the things that I have repeatedly come across that students don't get and that need to, that they're kind of not what you might immediately think are holding students back, but as soon as so if you can get the point over, it opens up a greater understanding. One of the things that on the stat side is that when you've got a sample of data, whether the top one there is nominal data, males, females, all along, and the bottom one is scale level, it's length of sperm in gorilla. You can, that's sort of its raw form, but you can also produce it in a frequency form, and that it's essentially the same data, just processed one level, going to a frequency. That just getting that idea over is really makes a real difference to the students. They understand suddenly when to use a chi-squared test more easily. And those things will build up, so when they go on in higher levels to do things like DLZMs and things like that, then they've got that more of a foundation of understanding the data. So it's unpicking misunderstandings and making sure they've got the key foundations for the higher level stuff as well. Next slide, I think that's my last one. And talking about that, just the undervalued frequency distribution and understanding what that can tell you about the data. So the frequency distribution is just on one sample, and it's not a frequency distribution like a histogram of continuous data is that's just one variable. Whereas it's not to be confused with a graph that's depicting the pattern or difference between two variables. And what it does is really help you understand your data. So quite an emphasis on that and the fact, and then that links obviously to sampling, because your bigger your samples, the more likely you are to be by theoretical distribution. And a little bit about theoretical distributions, again, laying foundations for learning more advanced stats, as well as, the toolkits don't go into any details of how to do various statistical test analysis. They're mostly laying the foundations of that. I think that's my last slide. So there's just a few examples to do that. So language, biological context, visualization, spotting gaps, particularly, I think Jenny's really brought that to the table with her experience of teaching in schools as the school curriculum, but both of us picking up those gaps or those quirks of things that are easily misunderstood from years of teaching. So I think that's probably enough from me. I just noticed that in the polls, that we've got still no one has ticked none. So perhaps people, obviously selected people attending this webinar possibly are already anticipating or would come with a particular interest in that. But it is something that we hope that the approach that we've taken and the evidence of our evidence is that it will address lack of confidence, because students will suddenly see things that they won't, by looking at things and learning the language, they will actually be able to unpick it. So it'll build confidence because they'll be able to do things they couldn't do already. It will help with their motivation and the lack of the skills, because we're picking it up for a slightly common level, a lower level of what you would. We're not expecting someone to have done A-level maths when we write in the toolkit. We're starting from a C grade in GCSE. So we think that these things address those kinds of challenges. Anyway, I'll hand over back to John. He's going to, I think, Stephanie. I'm going to do Stephanie. Stephanie, are you there? You're going to join us and maybe or give us some questions if there are any. Otherwise, I can keep talking. Jenny and I can keep talking about this forever. So
yes.
No, it's been incredibly interesting to listen to you both talking about it. So thank you for providing so much detail about your experience and various things that you've come across trying to teach maths to biology students. I'd like to encourage everybody listening to submit your questions. OUP has prepared a couple of questions that we can ask Dawn and Jenny to continue the discussion. But please do add your questions if you think of anything you want to ask about. You can add them into the chat and we'll pick them up. So we have actually got a question from Mark Edwards, who's listening. He asks, do you cover R, and is there any support for modelling of data?
Okay, so there is support for modelling of data in the very fundamental idea that students get to know what modelling is. So, for example, making them understand that if you draw a histogram, you've got a sample of data, and you draw a histogram of that, have you got that? Then you can model that with a mathematical line of the normal equation. So understanding that what models are, that they're the smooth lines on the graph generated by mathematical formulas that trying to fit the graph, and what that and the kind of things that can tell you. So for that example, it would tell you what you can use, parametric statistics if that happens. So modeling, we do cover modeling in a sort of really ground root kind of way, which hopefully that would lay so the students will then understand building up when they actually do specific techniques. So to ask the other part of your question, do you cover R? Well, one thing I'm quite passionate about is separating techniques from tools. So in this, we talk about techniques a lot. We don't talk about, in the stats side, I don't specifically give the recipes, if you like, for any particular statistical software package. So so the idea that students can calculate a mean and do it appropriately and report it appropriately on an appropriate set of data, that's where the toolkits are coming from, where they can choose a t-test appropriately. Then doing that t-test is not something that we deal with in the book specifically. There will be some online help to conduct the test in R and SPSS, but not. But that's not primarily where we're going.
Can I just add into that too?
Yeah.
So, I think that question was I assume that question was largely about statistical modeling of data, but I think it's worth pointing out that we do cover mathematical models to an extent in sections on equations, in formulas and equations, and in rates of change toolkits, where we talk about exponential growth and exponential decay and Michaelis-Menten equation, and thinking of those equations as mathematical models and connecting up the biology with the symbols and the graphs. And that is another form of mathematical modeling. I'm not sure whether the question was getting at that, though.
Well, I think it's important to, I think it's really something I find amongst, it's grown over the last 5 to 10 years, as R has grown in popularity, and I'm a big, I totally think it's great. And, if it's free and all of that sort of thing, and it's so flexible, and the community aspect, you can do anything. So R, I don't downplaying R, but there is a tendency for students to think, because they've done something in R, they understand it. So they pop out something, the mean of a set of data in R, or a graph in R, and they think if they've done it in R, therefore they have, they know what a mean is, but you can't do a mean on ordinal data. So that's the kind of point in the process that we are focusing on the toolkit. Sorry, Stephanie.
That's all right. No, not at all. The next question, there's been a question from Ian Sherman, but it follows a question from OUP. So I'm kind of going to give you both at the same time. So bear with me. So the question from OUP goes back to the statistical stuff again. And the question is, what statistical packages would you suggest students use? And then the question that Ian Sherman has asked on top of that is, following on from Stella's question, open source software such as R is starting to dominate the life sciences. Do you think there is a danger that students are starting to use these packages before they have a sufficient grounding in statistical data analysis?
Yes, yes, absolutely. Well, I think colleagues are, they're so enthusiastic about R and that, and they. I actually had an interviewed a student not long ago for a PhD, and I said, well, what techniques would you use to analyze this data? And they said R, and it's very much in the mindset of people that. Yeah, but yes, I would totally concur, and that's one reason for not. There's only so much you can do in this textbook. I have to say, in biomeasurement, the other textbook John mentioned that he commissioned many years ago, that does go through. Actually, the textbook itself has recipes for using SPSS, and on the online stuff has recipes for R and SPSS. But the focus of the toolkit is trying to, hey, hang on, let's make sure we're doing whatever we do is on the right thing. So I think, and pragmatically, in terms of what statistical software should be used, I think there's no doubt that for your research biologists, anybody wanting to become a research biologist, the sooner they get into R, the better. Anyone going off to who's not from the UK and not like won't go to an employer which has a statistical pay for a statistical package, then R is free, so they can continue using the same package. So there's lots of advantages for it. However, there are lots of, in terms of employability skills, there are lots of students that will go on to other areas, and I think that there's a lot to be said for, not just teaching one package, because actually what you want them is to know what a package can do for them and know how to teach themselves those skills. I think that's where we should be looking at. Yeah. So I stopped.
Yeah, that's fine. No, that's brilliant. Thank you, Dawn. While we're waiting for more questions from people listening, we have another question from OUP here to ask you. Do you think that maths and stats should be taught as separate modules or courses?
Oh, that's controversial.
So can I? It's all right if I answer this one. You can correct me afterwards. So I think that meaning that maths and stats should they be? I think, well, I should tell you what we're doing. We do at ARU. So we teach the maths and stats in the first year, and now as a module separately from the other subjects. And originally, the book that John mentioned, the biomeasurement book, was just a stats book based on a course that was just stats. So we're successfully teaching stats in a separate course, and then now we're teaching the maths and the stats. Now, I have mixed feelings about that, even though it's been, quite successful, and I get, a lot of things have come out of it. I worry that it compartmentalizes the maths and the stats from the other parts of the curriculum. The reason I have a lot of pressure from colleagues saying, well, let's start our second year course in quantitative skills, and I am the one resisting that. So the way we're going now is we've got this first year module, which is quantitative skills. It's called Quantitative Tools, Techniques and Tools for Biology. We cover maths and stats and Excel, SPSS and R. So we've got that happening, and then I am actually going into other modules, and my colleague who does the maths at ARU going into other modules. So we have what we call QTTB, which is the acronym of the module. QTT meets ecology, QTT meets physiology. So I've resisted that pull to actually develop more specialised modules in the quantitative skills, and actually going into those modules and creating that link. And then we've done that a bit with the modules, the other parts of the textbook. The authors have come to us and we've worked together on it. So we have the quantitative tools kits, which are separate, but we've also delved in and helped with the quantitative side when they've appeared in the other modules.
And that's been, I thoroughly agree, Dawn, because I think if you have it as a separate, if you have the maths and stats as a module separate to the biology, or if you have the stats separate to the maths, separate to the biology, I think you're just at risk of not making all of those links and not embedding. The risk of putting the maths in with the biology, I think it can increase motivation and confidence, and they see the relevance and everything. The danger is if you try to do too much at once. So I think if you're going to be, so they need to be well supported. If you're going to be integrating the maths into a biological module, I'd argue you need to have small pieces of, that's why we've tried to do small pieces of video, where you just take this mathematical thing, this little nugget, and you deal with that little nugget of a thing. You've come from the biology, you've gone to your little nugget of maths video, and then you've gone back to the biology again. So you're never too far away, so it's still integrated, but you want to be wary of overloading too much new stuff, so just real cognitive load issue.
Okay, so I have to say, while I've had colleagues like Kate, let's do second and third year modules on quantitative skills, I've never had a student ask me, well, I may have occasionally had a student to ask me, but very rare. Most of the students would much rather see it embedded in the other modules. So going, part of the pull to do separate modules is from staff. I actually think one of the not a very good reason for doing a separate module is, but it is maybe a consideration is if you're auditing your course and you want to show that you do quantitative skills and you want to tick boxes, then having a separate module where you can say, look, that's where we do all these things, that's useful. But otherwise, I can't think of huge reasons for making it. The way we're doing it at ARU is that we, so all the resources are in that first year module, so we can link back and then have access to them. So we make a load of use of that. So it is useful to have a separate module in the first year, but I think beyond that, it's definitely not very popular with the students, in terms of what they came to university to expect to do, linking in with one of those issues that we've raised. Okay,
right.
So
don't apologise, that's great, thank you. We have another question from Ian Sherman, who asks, has the gradual reduction in practical fieldwork in university biology courses over the last 20 years impacted negatively on students' confidence with and understanding of data and its mathematical statistical analysis?
Yeah, it's a really interesting question. I don't think I'm in a position to answer that. And
I think, yeah, so
it definitely impacted their ability to do certain practical skills and collect the data and have that continuum from the data to themselves and actually work on real data and obviously ecological data, which is quite messy. I'm not sure. I think that's more where the impact has been rather than on. I don't think that's been a negative impact on the quantitative skills side. But Jenny, you may have a different.
Yeah, I don't think so. And I think actually the last couple of years with the pandemic, looking at what some people have done with the way they deliver practical skills or practical aspects of the course, I think is actually a real benefit. So, you know, there's been a big emphasis, certainly from some people I've seen, on doing pre-labs and making sure the students are really well prepared for the lab before they get in there. So, you know, reducing the cognitive load that way, taking it a step at a time. And then thinking carefully about when they're in the lab or when they're in the field, making sure that really counts and that they're only having to deal with, you know, the collecting of the data at that point. And then having a post lab where they go through things really carefully. And I think having access to lots of small bits of video, because maths is like, you hear somebody talk about it and they explain it and it makes a hell of a lot of sense. It's marvelous. And then you go away and you get a problem and you realize, oh, actually, I didn't really understand that, did I? So then you need to come back to something where you go, oh, yeah, I remember that module I did before, or that bit of that book or whatever. And then you can go, okay, let me look at that again. Okay, I see now. And then that embeds it much more strongly, much more deeply. So there's like a strength or a depth of understanding that's being developed. And I think
I'm looking at Ian's question actually. Now I've read it. He's got data in big letters.
Yeah.
And actually, actually the data part of it. So I think with exposing students, because you can compensate the field. I'm not sure I would link the practical field work to. That's the source of the data. So it would be really interesting to see that, because absolutely working on real data and thinking about real data is what they have to do. Whether that the university reduction in field courses impacted that, it will have impacted certain kinds of data. That's a very interesting question, and it is very much about the more they can be exposed to data, different types of data, and thinking about that data. So it's actually an interesting point.
There's so much big data now, so much publicly available, so many big databases available to get your hands on. I think there's massive opportunities.
Yeah, there is a lot of data. It's just that the student collected field data
does that make a difference?
Yeah, so that kind of data. It's not necessarily an issue in terms of generally understanding. They can apply the principles about the structure, the features of the data, the structure of the data. But actually, if they're ecologists, they're going to more easily, if they've got that sort of data. So,
Okay. All right, thank you. And apologies if I didn't read that question out.
No, it's me. I think when I read it, I saw the data in big letters, which were, made me realise that Ian was actually, to help me focus.
Brilliant, thank you.
Got too much oxygen, going up there.
We have another question from OUP, if that's okay. We've got a few students who are dyscalculic or dyslexic. What would you recommend, and should we adapt our teaching?
Okay, so I'll take this one. This particular interest of mine, I guess. So yes, so dyscalculia, students really struggle with number sense, and they often have strategies that they might have developed at school to help support that visual sense of what number. So it's the sort of thing, you know, we take for granted when you're little and you get dominoes, or you count things out and all that sort of thing you do in primary school to develop your sense of number. So dyscalculics will often struggle with that. So providing visual ways of representing numbers, which is what we try to do with some of the visual support that we use. And the other aspect of dyslexia is being really careful about setting out of mathematics. So having space around an equation, always having, if the equation is like a fraction, you know, with two terms, and I like that they are vertically arranged, not like slash, if you know what I mean, like A/B on the one line. Don't do that. Put A over B always, and have white space around it. So I'm afraid I think we're a bit of a pain in the neck, weren't we, Stephanie, with getting the typeset just right to try and set it out clearly. Because I think, you know, if you train to teach maths, one of the things you learn is how to present the working out on the board, on the whiteboard, and that visual setting out is really, really important. And I often have to stop myself from just scribbling and make myself write clearly and neatly with lots of space if I'm just working on a whiteboard with a student. So yeah, so we've tried to be as dyscalculic friendly and as dyslexic friendly as we can. But yeah, I would really appreciate any feedback if any others are interested in that too.
And thank you. And just to reiterate, not a pain in the neck at all. Accessibility is something that, you know, we're taking very seriously at OUP, as everyone is at the moment. It's a big topic that people are talking about. So no, this is super important stuff. So thank you for explaining. We still have a little bit of time left. So if anybody listening has any further questions they'd like to ask, please do add them to the chat whilst we've got a last couple of minutes in the webinar. But we have a couple of questions from OUP just in case. So I'll ask one here. Looking forwards to the future, in the next few years perhaps, do you anticipate any further changes in how students deal with maths in biology? And what can we be doing to support students in light of these changes?
Well, you've got the stuff coming through, the things that the changes to the math curriculum, the core maths and stuff that was happening across the UK, and that should be feeding through. We should be looking at more prepared students over the next years. I mean, that should be happening. It'll be interesting to see if it does. So, yes.
Yeah, I agree. I think with the changes to the maths GCSE in the last round, those students will have fed through already. So they should be in their second year now or thereabouts. But the main changes to GCSE maths really are around the ability to problem solve. There weren't really very many changes in actual mathematical content. It'd be really interesting to find out. I'm not sure how many people are doing. It'd be a really interesting study to do to see to what extent that's changed what students are able to do. But I think with the pandemic, you know, how is that? We don't know how that's affected. I mean, we know it's affected the teaching, and we know there's a greater degree of variability. But I think the other thing I would say is about core maths. Universities need to be thinking about maybe not requiring it, but strongly suggesting it's a very good thing. I don't think core maths, you know, it's alternative to maths A level. It's maths that is the sort of maths we've been talking about here, but in real life settings and proper problem solving settings, and it really develops their problem solving on from GCSE. So, you know, if you have any influence in your admissions in your institution to get people to really encourage uptake of core maths, it is getting much more common, much more available for students to choose in sixth forms now and further education colleges. So hopefully it'll continue to build. Yeah.
So when we met back in 2010, one of the in Reading at the ATA BBSRT workshop, one of the things that came up was that, you know, people talking about was the lack of logs, and that's at GCSE, and that still hasn't changed. So that when they revised the curriculum, that didn't change, did it? So there's still no logs. So if you are taking on students with just GCSE maths, they don't have logs, and I find that that's been a thing from the challenges from the staff side, is that, you know, you have a student who doesn't understand something like logs, and you think they should understand logs, and if you give the facial expression of, well, you should understand that, even if you know you give the right words, if your facial expression is, God, you don't know logs, you know, then you can get into this downward spiral. So in terms of confidence and building confidence, that understanding the gap and understanding the prior knowledge, what the students have got, is actually very important. So hopefully, you know, by setting it where we picked it up from, that will help with that, and hopefully also encourage staff to realise that's where they need to pick it up from. They can't assume these things.
Yeah, and I have to say, some of my colleagues, you know, when I start explaining things that seem really, really basic, they almost look at me as if to say, "Gosh, you're dumbing that down." You know, you're I don't know. They sort of look down their noses at me sometimes, and I feel really quite strongly that that's not fair. And these students, they are where they are, and we need to meet them where they are, not complain that they don't know the things that we think they should know. They don't know it. It's not their fault they don't know it. So I think if you're unsure, you know, looking through the level at which we're pitching these quantitative toolkits, I think will give you a reasonable idea of, you know, so some students will look at some of the toolkits and think, oh, that's really easy, I know that, and that's fine. But there will be a good proportion of students who will be looking at that going, oh gosh, I'm glad that's there because I didn't realise that, or the equivalent. So I think it's more important to make sure you start from an acceptable level and then move them to where you want them to be from there. But there's nothing worse when you're doing maths. I've done it myself, where I've walked into a seminar where somebody is talking about, I used to do a lot of mathematical modelling, someone's talking about maths that's just that little bit too advanced from where my experience, and you're just bamboozled. It's just like that somebody's talking another language. So, you know, I think we've probably all been in that situation where we know what it's like to have somebody talking about maths that we don't really follow what they're on about.
And that just feeds into the lack of the anxiety and the lack of confidence. Although maths anxiety, actually, there did some work in the States where they found that parts of the brain light up. There are people where the parts of the brain light up that have phobias, that if you've got a maths phobia, the same part of the brain lights up as if you have arachnophobia and you see a spider. So that's a more specialised kind of issue, which perhaps the toolkits don't address, but they address the more general worry, concern, less physiologically based anxiety.
Thank you both for engaging in those questions. There is one last question from Ian Sherman, but I'll ask you to maybe follow up separately if we can make that work. But I'll pass back to John now to wrap up today's webinar. Thanks, guys.
Thanks, Steph. Yeah, thank you. So I want to say big thanks to both Dawn and Jenny for taking the time today to share your insights. Really much appreciated. Thank you, Steph, for your masterful facilitation just then. And thanks to Stella and to Katie and our marketing team too for making the event happen. And of course, thanks to you all for being part of the session and for your valuable contributions and questions too. We'll look forward to sharing more details about our new project in the coming weeks as we approach publication this summer. But for now, though, thanks again and enjoy the rest of your days.
Thank you.